Today's lecture is on genetics and breeding in agriculture. This is part of the Agricultural Technology subject ALM 207, which is offered as part of the agricultural degree at NMIT. My name's Dr. Nikki Cooley. In the last lecture we talked all about the Green Revolution, this technology and how agriculture benefited from it. Taking this one step further, new technologies were developed which enable the manipulation of genes which allow for the modification of animal and plant genetics. The improvement of these genetics have allowed significant advances in agriculture. As part of this topic, we will separate the subject matter into several lectures. We will talk about the technology in plant and animal breeding, both modern and historical, and then we will go on to talk about genetic engineering. We will start with a definition of genetics and breeding. We will talk about the role that genes play in breeding and the role of genetic mutations. We will look at some depth of traditional breeding, both the history and the background. We will then concentrate on some examples of both plant and animal breeding. The last part of the lectures will be finished with breeding strategies based on genetic manipulation, the advantages and disadvantages of these technologies, and why we need this information for future and climate adaptation. So let us start with the definition of breeding. Breeding is when you change genetics to improve desirable traits. Breeding has been conducted on both animal and plants. In fact, humans have a very long history of breeding with animals and plants, and this began at the start of human civilization. The breeding of both animal and plants to improve genetic traits is a very desirable characteristic for humans. We do it for many reasons: for food security, to increase economic returns so we can increase yields or economic traits in our crops, to reduce the environmental impact of agriculture, or simply to meet consumer needs and trends. The manipulation of breeding by humans began with plants, and it is estimated that we have been selecting for desirable traits from about 9,000 to 11,000 years ago. Initially, the requirement was to select plants that had desirable food traits. These traits were kept and later enhanced by keeping and harvesting the seeds of these plants. The sophistication of breeding was significantly advanced by the work of Gregor Mendel. Mendel was a monk who lived in Germany, and he demonstrated that the inheritance of certain traits in pea plants follows certain patterns. This is now referred to as the laws of Mendelian inheritance, and we are going to talk about these in the next couple of slides. In order for you to understand Mendelian inheritance, it is first important for you to understand how to cross plants, so that you can get the desirable traits that you want. The first thing to do is to familiarize yourself with a flower anatomy. On the slide here we have a drawing of a flower anatomy. These contain both male parts and female parts. The male parts are the anther, filament and stamen, whereas the female plants are the ovary. Select two ideal plants with desirable traits you would like to cross. Remove the stamen of your parent plants. This stops self-pollination. Do this just as the stamen starts to open. Using tweezers, gently rub the fresh cut stamen against the seed plant stigma, dusting the stigma with pollen. Using your fingers to gently fold the flower's petals over the stigma. This will give the pollen a chance to fertilize the seed before the wind or the insects disrupt the pollen. It is always good practice to label your crosses. Once the seeds have matured, they are ready for planting. Once the seeds have germinated, you will be able to see the physical traits of your crossing. It was this simple process that Mendel conducted with his pea plants. He did many pea plants and he crossed many to get the traits he was after. In Mendel's crossing, he observed changes to the following traits. Seed shape, they were spherical and dented. He observed differences in seed colour with his crossing. Some seeds were yellow and some seeds were green. Flower colour, he was able to show some flowers were purple and some were white. The pod of the shape changed, as well as the colour of the pod. He also observed that the position of the flowers could change, axil and terminal, and also the height of the stem could change, and thus he was able to produce tall and dwarf varieties of pea plants. Mendel counted the number of times these traits occurred after each cross. It is perhaps difficult for us to understand in this day and age what a significant advancement that Mendel made in genetics. He was able to describe the pattern of expression by observation only. Be aware he did not understand the mechanism as we know currently today. That is, we understand the role of chromosomes and genes. Mendel discovered 3 laws of inheritance. The first law, the law of dominance. The second law, the law of segregation. And the third law is the law of independent assortment. In order for us to really understand Mendel's inheritance, we must first deal with some of the terminology. The technologies and terminologies in genetics have evolved greatly, and the use of language is very important in the understanding. I'm going to start with the word genotype. The genotype is the genes present in the DNA of an organism. They are usually denoted by a pair of letters, and this represents one genotype of one trait. You always see a pair of letters due to the mechanism of sexual reproduction. If 2 letters are the same, the trait is referred to as homozygous. If different letters donate the trait, then it is called heterozygous. Other terms which describe homozygous and heterozygous have been used. For example, a pure genotype is actually a genotype that is homozygous. A hybrid, however, is a genotype that has a heterozygous. The next term I'd like to deal with is phenotype. A phenotype is how the trait is physically expressed in the organism. This is perhaps the easiest of all the terminology for us to understand. For example, the phenotypic characteristic of my carrot is that the carrot is purple. This means that I have genetically modified my carrot to change the colour of the carrot from a traditional orange to a traditional purple, and therefore the phenotypic characteristic of this is the colour. Another example of this is described as the dwarf mutation has a reduced internode length as its phenotypic characteristic. What this is saying is that I have genetically modified my plant to reduce the internode length, which has resulted in it being smaller. This is the phenotypic characteristic, the reduced internode length. The next term I'm going to talk about is called alleles. This can cause some confusion, so I'm going to take this quite slow. When describing the genotype, 2 letters were used, as described previously. One of these letters comes from the female and one of these letters comes from the male. An allele is the term used to describe one of these genetic codes of these traits. Therefore, we can say that 2 alleles make up one genotype trait. If this is our example of our genotype, and our trait is represented by a P, one of these P's comes from the male and one of these P's come from the female. The genetic code that produces this trait is described here as AUTTAUUUU, and it is the combination of all of these codes that gives us the allele. You will notice that both of these alleles are identical or homozygous, and therefore both are the same. The technical definition of alleles is that an allele allows for a trait that is located at a corresponding position on the homozygous chromosome. For the online students, please complete the following quiz before you commence the second part of the lecture. It is important that you understand the terminology correctly, otherwise explaining Mendelian's inheritance will be very difficult. Please answer the following questions. Quiz 1. Is the following trait, represented by the letter S, described as homozygous or heterozygous? SS. Question 2. Is the following statement true or false? If a genotype is called a hybrid, it has a heterozygous genotype. Question 3. Which of the following is a possible abbreviation for a genotype? TC, YR, PP, or G? Question 4. What is the best way to determine the phenotype of a dwarf plant? A. Examine the pea's root system. B. Measure the pea's internode length. C. Analyze the pea's DNA genes. Or D. Examine the pea's proteins. Question 5. How many alleles for one trait are normally found in the genotype of an organism? Is it A1, B2, C3, or D4? How did you go answering the questions? The answers are as follows. Question 1. The following trait, represented by two large S's, is homozygous. Question 2. The statement, if a genotype is called a hybrid, it has a heterozygous genotype, is true. Question 3. Which of the following is a possible abbreviation for a genotype? The letters PP. In all the other answers, there are either not enough letters or the letters are not the same. Question 4. Why is the best way to determine the phenotype of a dwarf pea? The best way is to measure the pea's internode length, as it's the internode length which is the phenotypic characteristic. And question 5. There are two alleles for one trait that are normally found in a genotype of an organism. One allele is inherited from each parent. There are two parents. Please continue with the rest of this lecture if you were able to answer these questions. If not, please review the lecture. So now that we've understood some of the terminology, let's look at some of Mendel's inheritance. We're going to start with the law of dominance. While Mendel was crossing or reproducing his pea plants, he did this over many generations, and he noticed some interesting patterns. For example, when he crossed tall plants with pure short plants, all of the new plants in the first generation were all tall. He showed and recorded this observation, as in the table. The parent plants, pure tall times pure short, produced all tall stems. Similarly, when pure yellow seeded pea plants were crossed with pure green seeded pea plants, he noticed that all yellow seeds were produced in the F1 generation. The same was true for other pea traits, such as rounded seeds with wrinkled seeds, and axil flowers and terminal flowers. In other words, what Mendel noticed was that when the parent plants had contrasting forms of a trait, tall trait versus a short, or green versus a yellow, etc., the phenotypes of the offspring resembled only one of the parent plants with respect to that trait. This means that one of the alleles is more dominant than the other. One allele is more dominant in a heterozygous cross. The technical definition of the law of dominance is as, in a cross of parents that are pure for contrasting traits, only one form of that trait will appear in the next generation. Offspring that are hybrid for that trait will have only one dominant trait in the phenotype. The following is an exercise to see if you've understood the dominance rule. From Mendel's results, in the table, can you determine which trait is more dominant? For example, the parent's pea plants, the tall stem was crossed with the short stem, and in the F1 generation all tall stems were produced. Therefore, what is the dominant trait? Go through all of these examples and identify the dominant traits before you go on to the next slide. You may need to pause the presentation. So here we have the results of the dominant traits for each of the observations that Mendel calculated. For example, the homozygous tall stem plant was crossed with the homozygous short stem plants, and in the first generation or F1 generation all of the plants produced were tall stems. Therefore, you can deduce that tall stems are more dominant than short stems. Please check all your answers for the seeds, pod colour, seed shape and flower position. It is the allele that carries the dominance of the trait. In genetics, the trait dominance can be represented by the case of the letter. So, when a capital letter or uppercase is used, this shows a dominance. When a lowercase is used, this shows a recessive characteristic, or less common traits. In the P example, capital T equals tall dominant, while lowercase t equals a recessive. When we look at these alleles, we can see the genetic symbol described. If you have a genetic symbol, uppercase T, what you're actually describing is a homozygous dominant. That is, both alleles are dominant. Where you have a genotype symbol, lowercase t, you are showing that it's a homozygous recessive. That is, that both alleles are recessive. A heterozygous, one allele is dominant and one allele is recessive, is shown by a capital T followed by a lowercase t. The following table sums up these symbols and descriptions for your reference. You can see the genetic symbols alongside the phenotype that is actually exhibited on the plant. It is worth noting that the only way the recessive trait shows up in the phenotype is if the genotype has two lowercase letters, i.e. it is homozygous recessive. So in the example of our pea plants, there are two lowercase t's. It is also worth noting that hybrids always show the dominant trait in their phenotype, and this sums up Mendel's law of dominance. The Punnett square. This is a diagram that can be used to predict an outcome of a particular cross or breeding experiment, i.e. it could show and demonstrate Mendel's results. It is used to determine the probability of an offspring's having a particular genotype. Here is an example of a Punnett square demonstrating the phenotype of green and yellow bananas. The genotypes are described as a capital Y and a lowercase y. The definition of a Punnett square is that the Punnett square is a tabular summary of every possible combination of one maternal allele with one parental allele for each gene being studied in the cross. Using the Mendel's tall and short p observations, we can show the outcomes of the crosses using the Punnett square system. In this example, we have a dominant allele, homozygous TT parent, crossed with a recessive allele parent. That is, the recessive allele will show short stems. You can use the following layout of 4 squares to show this cross. The first task is to place the parent genotype descriptions at the top and to the side of the box. As you can see from this slide, the capital T's are placed above the box and the lowercase recessive t's are placed to the left-hand side of the box. We can now use this arrangement, or the Punnett square, to fill in all possible combinations of the cross. The theory goes you take one allele from each parent, and the 2 alleles will give you the combination. So, for example, the capital T from the male crossed with the lowercase recessive t from the female will give you a capital T lowercase t, or heterozygous. This phenotype will produce a tall plant. I have filled out 3 of the combinations. Can you fill out the last combination of the offspring? Try to do this before you continue to the next slide. The last combination to fill in was uppercase T with lowercase t, uppercase T from the male and lowercase t from the female. This phenotype for the cross will produce all tall plants. We can use this Punnett square to investigate other crosses of parents. Let us take a dominant homozygous tall plant cross, or capital T by T, and cross it with a heterozygous plant, capital T lowercase t. Again, start by placing the parents above and to the left hand side of the box. Then place all the combinations, combining 1 allele from the male and 1 allele from the female. All of the offspring from this cross, F1, will have a mix of dominant homozygous and heterozygous genotype of capital T lowercase t. This will result in this generation of phenotype from this cross will all be tall plants. In this example, I'd like to cross heterozygous parents. These are parents with a capital T and a lowercase t, and they are both the same. Again, start by the basics. Draw the cross and add the male and the female genotypes on the top and the left hand side of the box. Then, taking an allele from each parent, draw the possible combinations. In the first top left hand box, the combinations of the dominant T crossed with the dominant T result in a dominant homozygous. What you will see from this cross is a mix of dominant homozygous, heterozygous for stem length, and recessive for stem length. The phenotypes for this cross will be mixed. That is, some will be tall and some will be short. Most of the plants, 3/4 of the population, will be tall plants, but 1/4, or a smaller percentage of the population, will show a short phenotype, described as lowercase t, lowercase t. And this is the basis of genetics and the modern world of genetics used for agriculture. In the next couple of slides, I'm going to set some examples for you to work through. For this example of Mendelian inheritance, I'm going to use this illustration of cats. The parent is a white cat and one parent is black. As you can see, the first generation they produce all black cats. In the second generation there are 3 black cats produced and 1 white cat. We can use the letters CC to describe the difference between the recessive characteristic and the dominant characteristic. When a capital C is used, the characteristic is a black cat. When a recessive characteristic is seen, or lowercase c, the resultant is a white cat. We can now use the Punnett diagram to explain these crosses. As before, if you put the parents at the top and to the left hand side, you can then work out all the first generations of this combinations. Our parents are a recessive cat, that's white, and a dominant cat, which is black. Once you have calculated the offspring for the first generation, you will see that the most common generation is a capital C and a lowercase c, or heterozygous for this trait. Now calculate the second generation, or F2 crosses, assuming that the parents are heterozygous. In the slide you can see that we have done this for the second generation. What you will observe is that 3 of the 4 offspring will have homozygous and heterozygous characteristics, but these will be displayed as black cats, as that there is a dominant allele present in all. However, 1/4 of the alleles will be recessive, and this will show up as a white cat. So these two diagrams, the first Punnett diagrams explains the observations of the first generation, while the second Punnett or lower diagram shows the observations of the second generation. I have set one final task to see if you understand how these crosses work. If you are unable to complete this successfully, please go back and follow and listen to the last few slides. If you are still having problems with that, please contact myself via email and or within the tutorials so we can talk about this. I'd like you to please describe using the Punnett systems the following crosses. These crosses are from strawberry plants. One of them is white and the other one is red. The parents are heterozygous for colour and dominant recessive for colour of red colour. The parents are described as capital R lowercase r crossed with capital R capital R. Please fill out the first one generation in the Punnett square. Once you have obtained the likely offspring, select any two parents from the offspring. Use these to cross for the second generation or F2. You should be able to answer what percentage of strawberries are white in the second F2 generation. So, how did you go? Here are the answers. The first Punnett square shows the offspring. As you are crossing a recessive with a dominant, all offsprings were heterozygous. Their genotype was red colour. You could select any of these parents and they would be the same. Once you've placed the heterozygous parents in the Punnett system, you can then calculate the offspring. You will note that 1/4 of the offspring will be dominant. 1/4 of the offspring will be recessive or white. 3/4 of the offspring will demonstrate a red colour. This brings us to the end of this part of the lecture. Now that we have done several examples of crosses across several generations using the Punnett square and been able to predict offspring, the law of segregation should be really quite easy to understand. The technical definition is that the formation of gametes, the eggs and the sperm, that during the formation of these, the two alleles responsible for the trait separate from each other. The alleles for a trait are then recombined at fertilization, producing the genotype of the traits of the offspring. And this is demonstrated in the Punnett square using our tall plants. We have a tall heterozygous parent crossed with a second tall heterozygous parent. As you can see, there will be 3 genotypes here, demonstrating 2 phenotypes. One genotype is dominant homozygous, one is dominant heterozygous, and the third is recessive or resulting in a smaller offspring. This is the law of segregation. The law of segregation actually occurs during the process of cell division called meiosis. Meiosis leads to the production of gametes, which are also called sex cells. These are either eggs or sperm. Sometimes the term gamemetrogenesis is used instead of meiosis. In summary, the laws of segregation state that when crossing heterozygous parents, this will always result in the following percentages of the genotype. 25% will be dominant homozygous, 50% will be dominant heterozygous, and 25% will be recessive. In our example of tall and short pea plants, this will result in phenotypes of 75% tall and 25% short. We can use our pea plants again to demonstrate the law of independent assortment. If you look at the genotypes of parent plants and say that a dominant allele for round seeds represented by a capital R and a recessive allele for wrinkled seeds is represented by a lowercase r. If you then represent a dominant capital G for dominant allele for green pods and a lowercase g for a recessive allele, which results in yellow pods. The table below shows the Punnett square for all the possible combinations of both parents and offspring. The parents are in white and red on the top and to the left hand side, while the offspring are shown in green and yellow. You will see that the combination of possibilities of offspring is independent. Therefore, the law of independent assortment has been demonstrated.